investigation 4.1, we are thinking about how matter and energy flow during photosynthesis. So remember, when we're talking about matter, we're talking about anything that has mass and takes up space. And when we talk about energy, we are thinking about usable power. So how does this happen during the process of photosynthesis? So the first thing we need to do is set up our lab according to the lab procedure directions. So in each of these test tubes, I have 25 milliliters of spring water. The next thing I'm going to do is I'm going to add 10 drops of bromoethanol blue into each of my test tubes. And remember, bromoethanol blue is an indicator. Indicator is something that looks for the presence of something else. So remember, I talked about how an indicator of how fast I'm driving is a speedometer, and an indicator of the temperature is a thermometer. So an indicator in bromoethanol blue is showing the presence, it's looking for carbon dioxide. And it changes colors when it senses carbon dioxide. So I'm going to put 10 drops in each of the test tubes to begin with. So remember, when we do scientific investigations, they need to be a fair test, which means we do everything the same and only change one variable at a time. Okay, so the next thing that I'm going to do is I want to add some Elodia, that freshwater plant, into test tube B and into test tube C. But before I do that, I'm going to add carbonated water to each of these. And I'm going to add 10 drops. And we can see immediately when I start to add the carbonated water, my indicator is showing, hey, I sense carbon dioxide in here. See how it's turning a different color immediately once my sparkling water gets mixed in. So I can see immediately that my indicator has changed, showing presence of CO2, carbon dioxide. And I'm going to mix it around. So my indicator is working correctly. I'm making observations right now of the starting color of each of those before I put them in the different conditions. So I am going to then place a stopper on A, because remember, this test tube isn't just filled with liquid, it's also filled with gas. And I want to control the gas that's able to come into and go out of each of these test tubes. So test tube A is going to stay just like this without the Elodia plant in it, and it is going to be placed in light. So I'm going to use my clamp lamp, and I'm going to put that in a light setting. The next two test tubes are going to have the Elodia plant in there because we want to see, can these two test tubes show me what happens with matter and energy in photosynthesis. So I'm going to get the Elodia plant down in there really well. And I'm going to get my other Elodia plant in C. Go in. tricky. Get my plant down in there. There we go. Now remember, when I'm making a fair test, I have the same amounts of liquids, but I also made sure that I did about the same size plants. And there, now it's down in there better. Maybe. And I'm going to put a stopper on both of those too to control those gases that I have inside of each of those test tubes. So you are observing right now the initial color that we have. And then we're going to be making some predictions about what color we think these will be changing as the process of photosynthesis takes place. So test tube A is, remember, my control, and it's going to go into the light. Test tube B is one of my variables, and it is going to also go into the light. Test tube C, however, I am going to place that one into a dark box, and it will go into a dark cabinet, and it will not have any available light to it to see what happens with the flow of matter and energy during the process of photosynthesis.